or you can access the cloud version, which is available here at posit.cloud. You get 1 GB of RAM, so things like third-party machine learning libraries might not work, like TensorFlow. And this is what it looks like. So this is the console. This is where you can do interactive code. And you can change how everything looks in tools, global options, and then you can change like which panes have which things. So here's the console. You can do line by line things like you could with any other IDE. Here are how variables are set. So you do variable name equal to value, or variable name with this weird arrow value. And this originally existed for compatibility with S and S+, but most people use it today. I think it's because of memory things or things with functions, but I just use it because there's a shortcut to do it. Here are the different data types. So there's numeric, which are integers and doubles. So you could do class and then whatever you're doing to see what class it is or data type. So numeric. Character, like Python strings, surrounded by quotes. Logicals, true or false, like bools in Python. And then factors. Factors are qualitative variables like good, bad, great, etc. And you have to set it like this: factor and then a character string or a number, I guess. And then there are also some less common data types like null, and then different dates. Vectors, lists, arrays, and matrices. So these are common things that you would use with data. So here's how you do a vector. You do C and then the different elements of the vector, and they print out like this. Lists are made with this list, and you do X equals some vector and Y equals some vector, and then it prints out like this. Arrays are like this. So you do data, then you specify the dimensions, and you can specify the names of each row and column. And they can be more than 2-dimensional since they're arrays, of course. So this is how you define it in 3 dimensions. You would have data, 3 dimensions, and then a list of names for everything. So you can see 2D, 2D, 3D. And then this is how matrices look like. You can define them with number of rows and columns. And if you want more information on any of these, in R, you can do help and then put something there, or you can do question mark and put something there, and it'll pop up in the help menu here on the bottom right. Here's the source console or the source pane. So down here's the console, and here's source, it's the top left. It lets you write scripts in various languages, not just R, but also Python, SQL, C++, HTML, and also what I'm using for this, which is R Markdown, which is like a combination between Markdown, HTML, and R. And some of these may require additional packages that it'll ask you to install, like LaTeX if you're doing stuff with math. So here's some extra things. There's a function that you can do a sequence where you have start, end, and step size, and it can be replaced by just start, end if the step size is 1. You can put parentheses around any variable assignment if you want to print it and assign it at the same time. It's like the walrus operator in Python if you're familiar with it. And you can use LaTeX in R Markdown, R notebooks if you know how to use LaTeX. So here's an example of some LaTeX and what it looks like in R Markdown. And then here are some extra resources if you are actually wanting to use R. I would recommend this list of books that parent company Posit makes available. Some of them are free, some of them aren't. But I've used stuff like R for Data Science and R Markdown, the definitive guide to learn about R. And here are some list of cheat sheets for specific libraries and packages that you might want to use. Like Reticulate is used for, it's a translation layer between R and Python. And then there's some stuff with like deep learning with Keras and TensorFlow, and some stuff with visualization with stuff like ggplot. So here's what R tends to be used for most of all. It's for data visualization and for data manipulation, wrangling, and all that. So I'm going to be using Tidyverse to show you just some samples of what it looks like. So this is a ggplot plot of a data set of diamonds, which includes variables like the carat, the price, the cut. And this is just the X values are the carat and the Y values of the price, and the specific colors are the quality of cut. So from fair to ideal. And this is from the MPG data set, which is just cars, their manufacturers, their miles per gallon, and their class. So here's a box plot. Down here are the 5 different manufacturers that I chose. And here's the miles per gallon. Here's the standard box plot of everything, and it's divided by class. So these can be very useful when you are trying to show some results of data that you've looked at. And then last second, I added some real quick things like standard things like loops, for loops, while loops, and how to install third-party packages and load them. So to install them, you do install.packages in the name. It installs it to a specific folder, and you can do different things. So I did reticulate, which is, again, R to Python translation layer, and tidyverse, which is what I used for these visualizations up here. And you can load them with library and then just the name without the string. And that's just to show that it loaded. It's just a real basic introduction to R, what you can do with it. Yeah.
Put this on GitHub and share it.
Yeah, I have it on my GitHub already, but I can share it with you.
If you have time, maybe you can video record it.
I can.
We are video recording this.
Are you?
Already? Yeah.
Okay.
Yeah, we can. I know it's very fast.
Yeah.
Yeah. Okay. Thank you.
Mhm.
Yeah. Oh, I see. Let me turn that to the power.
I'm sorry. Okay. So let me open up. This is an assignment I did in a class called Advanced Engineering Mathematics 2 at my college. So each of these like bundles of 3 is the same thing driven by a different equation. So here's the force. It's like the energy equation, and then you get the force out of that equation, and then you put it through a big long process. So they all have their own like nodes. And so this like folder, this piece is just for the force from that energy. And ODE, I don't remember what it stands for, but it basically, it's like an array of information, and it takes the both XYZ positions and velocities, and then it takes the derivative of each of those. So then it spits out the velocities and the accelerations. And this is sort of like the math that drives that. This green is like an explanation. And this is pretty much just like the driving that pushes it to move. And then this is making plots of that movement. And let me close these other ones so I can show you guys. We'll start with, I guess, the linear inverse. So as I use for loop to go through the Y range, which is pretty much just the size to make the plot. And this color jet is kind of like a neat touch that I added to make each new plot a different color. So this is making an array of jet, which is a bank of colors of 20. And then I increment it, I think that's the word, at some point in the loop each run. And so every time it plots a plot onto the graph, it makes it in a new color. And my play button is so blocked off. But if we run this, you can see it sort of running through that loop and adding a piece to each plot as it goes in a new color. And the naming of the colors is really messed up because I did this on a time crunch. It's probably the reason I only got a 90 on this report, but whatever. So that's the process. And then these are the results in sort of like a nicer format, like easier to see. I guess I'll full screen this. So these are the separation between, oh yeah, the thing this is plotting, I forgot to say that. This is a diatomic molecule. So it's the two atoms and they're vibrating. And so this is the position of them in angstroms over time. So you can see this is one atom and this is another atom, and they're both just vibrating. And each color is a different initial position. That was that array that it's going through. So for each plot, it like increases the initial separation and then starts over. So that's what these are going down this line. So this is a position versus, I guess in each color is the change in separation. And then the average separation between the two, or not average, this is just the separation, but I do have a plot for the average. And here's the average separation. And the thing I'm comparing this to is the Morse potential, which we already know to be the most accurate one. So that's the hollow circles. And then in each plot, the dark circles are these different equations that we're trying just to look at. And here is the separation over time for different initial angular momentums, I think, right? Yeah, angular momentum. So for this, I made a new for loop, and instead of incrementing the initial separation, I was keeping the separation the same and changing the angular momentum. So that's what these new plots are made from. And here's the period of those oscillations versus angular momentum. I think, yeah, that's all the plots I made. So we can call it a day at this if that's enough. I also have a research that I did if you'd like to see more.
How long did you spend on it?
We spent like a third of the semester to half of it. So like quite a bit of the semester on this project. And then we spent the rest of it on differential equations. Thank you. I also can run through very quickly the research I did last year, which is it's sort of like throwing a ball through a fence, is this concept. So this material is called biphenylene, and then these are different atoms passing through the largest hole in it. And then we're just looking at this for use as the filter in a battery. Just like a speed run version of that. That's just something neat. And all these pictures were made with a visualization software called Vesta. This is like this really sloppy version of this. This presentation is on YouTube, actually. I could post it in the chat if anyone would like to see me presenting it at Vanderbilt. So I can go ahead and wrap up and then send that after. That's good. I like color. I also have a presentation about carbon nanotubes that I made just for a class, and it also is like really pretty. I like to make things pretty. At the batteries. Yeah, that's what a lot of the stuff I've done so far tends to connect to is batteries.
Okay, so this is kind of what I was working on last night. It's basically just a form of battleship. Um, so I have two different classes. One is just a ship itself, how long it is, whether or not it's been sunk, and the location and how it's been hit, and whether it's oriented vertically or horizontally, and then the type of the ship for other things. I have two different class constructors for these. So basically, these will make a ship depending on the type of things that you put into it. One of them is for starting the game, this one here, and the other one is for if a ship were to move later in the game, which I have something for that. And then just a whole bunch of observers that give certain parts of the class that would be necessarily private.
This is a C code.
Yeah, this is all C++. A whole bunch of observers, and then down here, this function is basically whether the ship will fit in a certain spot or not, so it doesn't go past the board size. This is a function for checking whether the ship hits. This function is for checking if the ship is visible, which is another thing that I added for like a little extra thing. And then this is a comparison here for comparing, for ordering the ships, so that ordering all the points that each ship takes up. And then this class is for the game itself. It has one board that is what the player would normally see, and then another one that actually shows where the location of the ships are for testing purposes and stuff like that. And then it has a vector, which is basically an array that's dynamically allocated with a whole bunch of ships inside of it, and then the orientation so that they can be random. And basically what it does is it makes both of the boards that are just empty, creates a random seed, a pseudo-random seed based on the time, and then initializes a whole bunch of orientations for the ships, and then initializes the ships, which basically just adds a whole bunch of them to the ship's vector here. And then every time a ship is added, it adds it to this board for the cheat board where you can see where all the ships are. And then every time one is added, it uses this board here to check and to see whether like a ship placement is legal. And if it finds that a ship placement is not legal, it will then try and place the ship again. This function is for later on, where basically it takes the ships, keeps things that you've hit, and then changes the placement just to make it different, I guess. And it was a whole bunch of work to put this thing together, but all that it basically does is just move the ships around. This is a whole bunch of initializations for starting the game, just initializes the beginning of the ships. This prints the board every turn, so that you can see both boards when you play the game, and then this is the actual game function.
So this is basically the battleship for the game to put in.
Yeah.
The first function here is
So basically every turn, it will check a certain timer, and if the timer is 0, it'll move all the ships around, which is what this function up here was. There's a whole bunch of messages that it puts out, depending on certain variables and whether they're set. And then it checks to see if the game is over before it asks you to put stuff in, so that it'll terminate the while loop, and then it'll ask you to enter a row and column. Check and see if the move is valid, make sure it's not something you've already hit, add one to the turn counter, and then it'll check to see if you've hit an actual ship that exists, and then change the actual board to reflect whether you hit it or not. And then this is where the timer is set. Basically, if the timer is not 0, there's a 20% chance, I think, 20% that the timer is set every turn. And then once it's set, it sets this variable right here to true, which then tells you that a timer has been set up here. Yeah. And then chooses some random value to set the timer to. And then these are just game over in legality. And so if you actually run it, this is what it looks like. So on the left side here, this is the actual board that the player would see. And then on the right, that is where the actual ships are. So you have all 5. You have the patrol boat, destroyer, submarine, battleship, and the carrier. This line right here tells you which of the ships are sunk. And you can enter coordinates. This is a hit right here. As you can see, that corresponds to this patrol boat. If you were to put in something that's not a hit, it puts in this 0, which is a miss. And then this message right here tells you that at some point the ships will rearrange themselves. Yeah. And so this message right here means that I have exactly 2 turns before the ships will realign themselves. So if I just do this, you can see that everything's changed. And there is a percentage chance that you won't be able to see something that you hit. And then if you hit it again, it will tell you that you'd already hit it before. So that's all that I have.
Okay, so this is quite different from the other presentations so far, but let me see if I can.
You have a poster.
Yes, I do. Okay, so last summer I spent last summer in an organic chemistry synthesis lab, and then last semester I presented my research at my college's Scholars Day. And so basically my research project is the synthesis and characterization of meso-substituted phenothiazine, zinc porphyrin, zinc phthalocene diads. And so basically what I did with my graduate mentor was we created these porphyrin and phthalocyanine bodies that are displayed on the left side here. And porphyrins are actually found in our system. They help capture blood in our hemoglobin, but they're also found in chlorophyll, which helps in the photosynthetic processes. And then phthalocyanine, the structure also resembles something in chlorophyll. And so both of these basically help to mimic photosynthesis. And the lab that I'm in, we basically create these materials to convert light into like electricity, energy, fuel. And what you can do with these porphyrin bodies, you see that there's an R group right here. We can attach different functional groups. And so I have 3 examples over here. And we basically measure the amount of light that it can absorb. And so for my project, we put in 3 phenothiazine groups in the meso positions of our porphyrin bodies that act as electron acceptors. So when they take in light, they funnel that energy into the centers of porphyrin and phthalocyanine bodies, which then donate that energy into other molecules. In this case, we use fullerene, which is this like buckyball structure right here. And so that helps to initiate some sort of conversion between like other molecules. So we make a useful product out of that. And in the organic chemistry synthesis side, I basically did the synthetic scheme and the UV visible spectroscopy analysis portion of this compound. And so this is the synthetic scheme right here. It seems simple on paper, right? You put compounds together and it should spit out this, but it takes a long time to purify these compounds because you can't really move on into the next step without actually having the pure compound and a reasonable yield. And so we use things such as column chromatography and thin layer chromatography to separate these out. And then afterwards, once we do get the desired structure, we then run this in the UV visible spectroscopy, which is a machine that shines like a beam of light with a known wavelength through the sample. And so on this side, we're measuring the absorption values. And then on the right side, we're measuring the emission. And absorption basically means that the energy required for that electron to move from one energy level to another, and then vice versa. Over here, these are called titrations. And so we're basically doing the same thing that we did on A and B, but with increased concentrations of the fullerene, which again was this ball right here. And this just basically measures whether or not like our compounds are coordinating together or if they like act as independent sites. In this case, it does coordinate together, or else we would see different peaks. And then over here on the geometry optimization, this was run by an analytical chemist in the other side of our lab. So I thought this was really, really cool. I'm not well versed on the analytical chemistry side. They did use a program to create these structures, but this just shows the electron cloud. In other words, the space in which we can find electrons. And then over here, EGH shows the HOMO gaps, or the highest occupied molecular orbital. And then on the the right-hand side is the LUMO side, the lowest unoccupied molecular orbital. And so analytical chemists will measure the difference between these gaps to see whether or not our compound can absorb light. And so, yeah, that's it. Oh, and then I'm not well versed in coding yet, but I did manage to make my first coding game. So I'm really excited about that. I made Pong yesterday and I made it with sound too. I don't know if you
did this yesterday?
Yeah.
Oh, wow.
Yeah, I did this yesterday and yeah, let's see if it'll run. Oh yeah, it's here. I don't know if you guys can hear it. I put my laptop on mute so that I wouldn't echo, but it'll boing here and then it'll make like an error sound here. And if I press Q, it'll say good game and I just ended the game. And yeah, I made this yesterday. It was exciting. It's really cool. Yeah. Okay, thank you.
Did you?
No. So what I did was I found a full course Python tutorial on YouTube, and it was really long, but I did half of it. And then I was like, okay, let me utilize what I learned into a game. And so that's what I did. But I did ask ChatGPT how to terminate it because it didn't tell me on the YouTube video, but it helped out. So yeah. Thanks.
So I'm a biologist by trade. I have no computational background, but this is the research project that I did with my class last semester. So we were doing RNAi and C. elegans. C. elegans are these little nematode worms. They're about 1 millimeter long. They're used in as model organisms. They have like a short life cycle and they're clear, and they have various other traits that makes them really good model organisms, including for human diseases. So we were using them to study gene expression, like we were talking about earlier, the whole, DNA gets transcribed into RNA, gets translated into protein. Specifically, we wanted to study like how you can discover the effect and purpose of a gene by silencing the gene. You turn it off, and then seeing what happens to the worm when that happens. So we were doing it by RNAi, which is a process where you use double-stranded RNA to silence the gene. RNA is normally single-stranded, and double-stranded RNA is generally found in like pathogens. So C. elegans have developed a defense mechanism where if they detect double-stranded RNA, they'll destroy all the RNA that matches it. So if you give them double-stranded RNA that matches their own RNA, they'll destroy their own RNA, and then they can't produce that protein. So we were specifically silencing the gene TLN1. It's a muscle cell development protein. So it's one of the signaling pathway proteins that is involved with the muscles development. The muscles are found in C. elegans are found like along the sides of the worms. There's a lot of possible phenotypes that can come out of this in what stage they receive the RNAi treatment. So if their muscles aren't properly developed, they can be paralyzed. If it hits them really young, they can die as embryos. These embryos, larvae, adults as their life cycle. They can go sterile. They can lose their ability. They live on gels generally, and they can lose their ability to burrow into it, which they normally do when they run out of food. And then they can just not be fully paralyzed, but just kind of struggle to move. The main phenotype we were looking for was paralysis. So C. elegans worms, I should have pointed this above. They normally travel in this sort of sinusoidal pattern, and you can see it when even they're just resting. They have a little sinusoid pattern just when they're hanging out. But paralyzed worms don't have this. They kind of look like string beans, and they look a lot straighter. So you can often detect them even without like checking for motion. I mean, you want to double check with motion, obviously, but if, say, you're making a PowerPoint and you can't embed a video. So we ran several trials of RNAi, counted our worms and our paralyzed worms and our larvae and adults and eggs. And we did get paralyzed worms. The guy in the middle, as you can see, he's a lot straighter. He's a string bean banana worm, and he's paralyzed. And then the guy in the upper left, who still has the sinusoidal pattern, was not paralyzed. So we had 3 different treatments. We had our L4440, which is our negative control. And as you can see, there was, as predicted, no paralyzed worms. SPD2 was our positive control, but it wasn't positive for paralysis. We'll get to it later in a second. So it was also not predicted to give paralysis, and it didn't. And then TLN1 obviously was predicted to give paralysis, and we did get 52% of the worms on average were paralyzed. And we ran a t-test and found it significant. So then for our SPD2, which was our positive control, it was supposed to give the embryonic lethal phenotype, which you detect by taking the ratio of eggs to eggs over larvae, because if they don't develop to larvae, you're going to see more eggs in proportion. So we were supposed to have much higher SPD2 ratio than L440, but our trials weren't long enough, so they didn't have time to develop far enough through their life cycle for it to be detectable. So we didn't get a positive result for a positive control, but we did get a negative result for our trial, or the TLN1, the experimental one, which was not expected, and we were kind of confused by that. But it occurred to us that we could, if these worms were sterile, which was another predicted phenotype, it would have fewer eggs because they're just laying fewer eggs. So then we calculated the eggs per adult, and we found that they were much lower. So it was likely that the worms not only were getting paralyzed, but were also becoming sterile. SPD2 was also giving a fairly significant sorry, low eggs per adult ratio, which was a little confusing, and we sort of pinned as something to study more in future studies. Back last semester, we were doing a rather simplified version of the math. We basically were plugging stuff into Excel and just taking a bunch of averages. And one thing that stuck out to me is that we weren't doing it. We were counting all the different plates we had run as exactly as if they had the same number of worms, which for my group was definitely not true. We had a range from some had like 130 and some had like 20. So I wanted to recalculate them and sort of account for the fact that there were different numbers of plates in the worms. So last night I sat down and I started with ChatGPT and sort of got it to spit out some code and then went from there. By the end, I was doing a little bit of putting in my own, using the earlier stuff as like a template. So I recalculated it, sort of pretending they were all on one plate. And I did get somewhat different answers. The lower numbers are the different answers. So 52, 50 was not that big a deal. The big one that stuck out is the ratio of eggs to eggs over larvae for TLN1 is definitely higher. So that's something we'd want to look into more if we were redoing this experiment. And then it's the eggs per adult was lower for SBD2 and a little higher for TLN1. So overall, we did see the expected paralysis phenotype and possibly also evidence of sterility. SBD2 was being kind of weird and we bookmarked it for future studies. And overall, our hypothesis was supported. And then lastly, it's neither mine nor really research. I was talking with my brother who has a lot more experience in Python and he made a bunny. So I wanted to show you guys that. Yeah. And then, overall, I just basically been trying to teach myself the very basics of Python.
So you talk to your brother online. Is your brother older or younger?
Older.
Oh, what does he do?
He teaches second grade.
Impressive.
No, it's no, it's just a lazy citation.
Okay. I saw you very practical. All right. So the books that I need to
Hey, David and Skylar, do you have something to share?
I can share something.
Okay, great, thank you.
Hey, just give me like 2 seconds. How do I share my screen?
There's a green share screen button at the bottom.
I'm sorry about that. Yep, just give me 1 second. Okay, is it working?
Yes, we saw something on your screen. Yeah.
Okay, is this, I think this is, I can't tell if this is my entire screen or not. Sorry. I've never done this before. I've never screen shared before, so give me like 1 or 2 seconds to figure this
Yeah,
out.
we saw something, National Library of Medicine.
Yeah, you can see this, right? So
Yeah,
this
we
is,
see some, yeah.
so when it comes to computational biology, I haven't necessarily done much that associates directly with biology, but this is probably the closest. I think I mentioned in my introduction yesterday that I'm doing a machine learning project in a like with data from NIH, and this is well, basically I sampled specifically the AMR genotypes. And after I downloaded that and did some like copy and pasting, I essentially ended up with 2 columns, which is, oh, and I just messed something up. Okay, which is the strain, which, wait, I keep messing something up. Sorry about that. But one column is the strain, which these are all strains of E. coli, and the other one is the AMR genotype cores. And so the issue at hand was essentially there's I have no idea how 6,307 of these, and I had essentially, well, if I enlarge like all these genes, you can see that there's quite a few, and like obviously each strain has different ones. So essentially what I had to do is I created a program here with AppScript, which basically like took these values and created a column, because a lot of these repeat, and created a new column every time a new one was introduced in every single row. And it would put a 1 or a 0 whether or not it would say complete or point, or I mean obviously if it's just not there at all, because all I needed to do was like know if it was complete or not. So as long as that was there, I could just put a 1 instead of a 0. So with this, well, I mean, I kind of just already explained it, but it recognizes sorry, it reads if there's whatever gene it is, and then it puts either a 1 or a 0. And the reason why it needs to be in this format is if I were to save it and in the in .csv, it would put it in the correct format, which I need to use in I think, oh yeah, in Spider, in order to for my machine learning, but I haven't gotten there yet. So this is like as far as I've gotten. I used ChatGPT a lot to help me with this code, because I'm pretty sure this is in JavaScript, which I don't really know at all. So that helped me a lot. Whereas once I actually do migrate this code, this data into my machine learning thing, it would be in Python. So those are different languages, but this is probably the only thing I have that's most similar to computational biology. However, I have another coding thing I have pulled up, which is this little game I made last semester for my one of my projects. And this is just in Java, right? And essentially I don't know how familiar everyone is with Greenfoot, but specifically like it's pretty good for making like really basic games. And this is just a really basic game where you catch fruit. And essentially with this, I was just trying to think of a way to make a really simple game, like have like a couple like more complicated aspects to it. So obviously you can see at the top left, there's like a coins counter and then a timer. So there's 5 levels, and each time the timer is 20 seconds for each. And if you survive to the end, you have 2 options. You can either go to the next level or go to the shop. And if you were to go to the shop, you can, this is actually incomplete. I haven't added this function yet, but you can either buy a cosmetic and change the appearance of the bowl for catching fruit, or a lottery ticket. And then there's a button here that allows you to go to the next level. But oh yeah, I wanted to show this off because a bunch of these, like a lot of the code here it's like it appears extremely simple because the outcome is extremely simple, but the way to get there is just like needlessly redundant and complicated. For example, in order to make the fruit that loads in look like it's coming from like the top of the screen, because this is supposed to look like an arcade machine, I had to implement like another sprite essentially that covers up the fruit that spawn in. But the issue with that is based on the order of the code I put in, that determines like what fruit what sprite overlaps what sprite. So the only solution I found to doing that was essentially every single time a fruit is generated, it would remove the bar that's already at the top and bottom and replace it for every single fruit that generates, so it would look like that it overlaps. Now oh yeah, there's another twist to it where one of the things you can purchase in the shop is this like little lottery ticket, and there's a 1 in 9 chance if you click one that immediately like either makes you lose or makes you win. And all of the fruit have different movement patterns. So it's like, let's say if I go to like level 3, it should be only oranges, I think, and they move in a diagonal pattern, which is determined by this code here. And I mean, yeah, that's about it. It's just a very simple game coded with Java. I could sit here and go through all of the code, but this is kind of, I just wanted to show off some past projects I've been working on. But yeah, that'll be about it.
Okay, thank you.
Wait, you could see the game I pulled up, right?
Yeah, we saw it again.
Okay, sorry.
Yeah. Okay, it's time to break for lunch, and so when we come back, there are a few students still left. I think Emma, Kaylan, and Tamara.
I'm so sorry to interrupt. How do I stop screen sharing?
You click the screen share button again, it will stop sharing. Hey, Skylar, are you there?
Yeah, I'm here. I have something I can share too.
Can you do that after lunch? Oh, you prefer to do it now? Oh,
I can do it after lunch. That's fine.
okay. Yeah, I'll see you after lunch at the 1. Yeah, bye. I'm going to stop the recording.